Yeah. Okay, well, thank you for the invitation. I actually started working on sarcomas in 1982, so it's quite a while ago, and did that until I got into the administration, which was in 2012. It's a pleasure to be here. I'll try to be brief. I have 29 slides, but I tested it and I should be able to do it in 20 minutes, so don't worry. And as Annika already told you, I am a medical oncologist from origin, currently working at the Cancer Drug Development Office. I end with the aims that the Cancer Drug Development Office has to integrate science and objective and subjective elements into the drug approval process. I found more definitions of evidence than Annika has found already. If you just go to the internet, you can find 27, but basically what it is all about is finding proof and finding what you could say the truth. Whether that's really the truth is also something you can still debate. You can debate forever, but evidence is something that leads to less discussion than subjectivity. So basically we go from belief or a proposition or an expectation, which is subjective, to something that nobody could debate anymore, but everyone accepts as the truth, a non-debatable certainty. If you go to scientific evidence, we go even one step further. In science, belief is called a theory, or it's called a hypothesis that somebody postulates. And that postulates or that theory has to be proven before we consider it actually the truth. And therefore, we have agreed scientific methods that we use to achieve that objectivity. The methods differ in the various professions and the various fields of research, but the essence is always the same. And what is important with scientific evidence is that we only conclude so if we have a rigorous, controllable data set where a statistical analysis has been done upon. So we use mathematics or statistics to prove to each other that the postulation or the theory is right. And the important element here is also that somebody else should be able to repeat exactly the same experiment and come to the same conclusion. Otherwise, you still start to doubt. So the rigor there is an essential element of what scientific evidence is. Basically, what we're trying to do is exclude the possibility of observation by chance. You can listen to the news currently with the vaccines, the potential side effects that have been reported. And then the question is always, is the side effect a true effect of the vaccine? Or is it an effect that just occurs in mankind? I mean, if somebody gets pregnant after vaccination, is that a result of the vaccine? Or is it something that just happens in mankind? So we're trying to exclude that possibility of chance observations. Now, if we go to clinical science, clinical parameters that are used for scientific evidence in oncology, this is not an endless list, but these are the most important ones that we are currently using. And I'll discuss those briefly. So survival is one thing. Tumor extent or tumor burden and the changes in the tumor burden is another thing. Symptom changes, very important for patients, of course, are another thing, and quality of life changes are the fourth thing. And there are additional ones that I'll briefly discuss in one slide later on. So let's go to the very first one, survival and tumor burden change. And I'm going to that first one because the other two are subjective. Changes in symptoms are subjective. It's perception of somebody, of a human being. And you can never prove as a doctor that that perception is right or wrong. Of course, for the patient it's right, but whether that means that it is a resultant of the treatment or a resultant of somebody else or something else, we will never know. It's very hard to objectively show that. So let's go to the less subjective elements and the objective elements of trying to get proof. And survival, of course, is one thing. People always say, well, there's basically one thing that you cannot fraud upon, which is the date of end of life, because that's listed. You'd be amazed that people can still take a fraudulent date of end of life. But basically, the end of life is something that we can measure because the date of death is unfortunately a very solid date, a very fixed date. Now, you have to have that date of death, of course, to be able to calculate what we call overall survival. And in this example, you can see that there are various stages in life that a human being goes through, and then that person is put on a treatment, in this case, a study treatment, which is the green part of the arrow. And the length of the green part, you could say, well, the start of the green part is where the treatment started. The end of the arrow is where life ended. That's what we call overall survival. That's measured from the start of the treatment until the date of death. Very objective. The problem is, if the patient is like this and goes on a study treatment at the beginning of a disease period, and then has other treatments subsequently because the treatment fails. I mean, Roger is one of the examples where many things have happened in his lifetime that I know of. And then measuring overall survival is much more difficult, since how much is the contribution of the initial treatment, the green part of the arrow here, to the totality of the total arrow? So their overall survival could be, even the total overall survival could be more a resultant of a subsequent treatment than of the study treatment that I made green here. In these cases, we need to find another solution to measure the actual result of the treatment that we give. And that's why people have started to use what we call progression-free survival, which means there is a disease, in this case cancer, you start to treat the disease, you see hopefully a good response to the treatment, but unfortunately at some point in time, there is progression. Progression means that the tumor extent is increasing again. The tumor is growing again. The period between start of treatment and the period between the regrowth of the tumor, we call progression-free survival. But there is an essential element here that is required to measure this. You need to be able to measure that extent of the tumor. You need to be able to measure the size of the tumor. Otherwise, you cannot assess progression-free survival. And in order to achieve that, we've always used criteria and language that enabled scientists all over the world to speak the same scientific language in assessing outcome of treatment. That was changed. We initially started with WHO criteria, and in the year 2001, we published a data set that was called RESIST. That stands for, the abbreviation is for Response Criteria in Solid Tumors. You can see in red the letters that lead to that word RESIST with a C in there. This was done by the NCI, the NCIC, and the EORTC. So this enabled every investigator in the world in clinical cancer research to speak the same language if we talk about responses. And survival, of course, is also a response. Importantly, if you talk about RESIST, you should talk about RESIST and not about RESIST criteria, as many, many investigators do, actually. But the C in the abbreviation already stands for criteria. So if you say RESIST criteria, you basically say criteria, which is nonsense. So let's talk about RESIST. Now, in RESIST, we have identified some definitions, and we have created some definitions, and we have given them a name. This is complete disappearance, complete response, or complete remission, whatever you say. The word complete there is essential. It means that all the tumor that we could assess prior to the start of the treatment has disappeared on treatment. Disappearance of all all tumor that we can see on imaging does not necessarily mean cure, but it's certainly a good effect, because some things we cannot see. So the disappearance of all visible, and in this case, clinically visible, radiologic or biologic signs of tumor, complete remission. We also have a partial remission. If you have something complete, you have something partial as well. And there we defined this as the decrease of the sum of the longest diameter by at least 30%. So the 30% is an artificial boundary, but it's something that we can use if everybody uses the same language again. And you see an example in the ovals that are on the left and the right-hand side of the screen, where you see that that tumor has been shrinking by more than 30%. So we call this a partial remission or a partial response. If something does not respond but grows, then we call it progression, progressive disease, but progression, it's basically the same. And there we have also, again, an artificial boundary, and that's 20%. So the 20% increase in the sum of the diameters, that's called progression. Why these boundaries? There's always uncertainty in measurement, and I'll get back to that in the next few slides. So this is what RESIST is all about. And it's very important that this has been validated. Scientific proof has been provided here that the system works. And this was done on a database. Initially, we started with 6,000 patients. Now it's over 20,000 patients in a warehouse of data that we can use in many different ways and show the world that this is really something that has rigor. And they can all repeat it on other data sets, so it's repeatable as well. Now, in order to have this progression-free survival, in order to measure the extent of the tumor, we need to be able to measure. And you would think, well, that's relatively simple. You have a CT scan or an MRI, you find the boundaries of the tumor, and you can find the largest diameter of that tumor. Well, I wish it was that simple. This is just an example of a thoracic tumor, and if I go to, can you see my arrow now? I cannot even see it myself. Yeah, here. Here's the tumor. And you can see there's a margin. Sorry. I apologize for this. Pushing wrong buttons. Yeah. Here's a boundary that you can see. This is air in the lungs, and this is tumor. This is not tumor. This is the heart. So we have to distinct the tumor from the heart already. And finding the margin here is relatively simple, but I can assure you there are instances that it's much more difficult to really find the margin where you could put your caliper. So when we were designing RESIST criteria, I did a test in 60 medical oncologists, all friends of mine, and I asked them to measure an exact circle. This was a circle that is exact. It's larger here, it shows larger on screen, but it was 60 millimeters. And I asked them to measure that. And then there are two circles on the right, and you can see just by eyeballing that those circles are smaller. Okay, the first one was 45 millimeters. 33% of my colleagues had it right. They could measure an exact circle in the correct way. 70% came close, and 23% didn't even come close. And the range in the measurement was between 41 and 52 millimeters, while the exact measure was 45. So, by definition, 45 millimeters is less than 30% decrease. So this is a stable disease. We call anything between partial remission and progressive disease, we call that stable disease. Now, the smaller circle there is 40 millimeters. 24% had that exactly right. 60% had it almost right. 40% didn't even come close, which shows you how difficult it is to measure an exact circle. And this is really less difficult than measuring something on a CT scan, which is lobular or whatever. So measuring is not easy. And my colleague Bruce Minsky in the United States did something similar. And he used CT scans. He gave CT scans to his colleagues. And you can see here that the tumors that were there are listed on the horizontal axis over here. And the measurements, the means are here. And the actual standard errors of the means are like this. So in a tumor that is 12.9 square centimeters, the standard error is like this. It's phenomenal. It shows the inaccuracy of measurement. And there are systems that can improve on this. With a planimeter, it's less inaccurate than with a caliper, but still it's inaccurate. So measuring is not easy. If we move to the more subjective topics of measurements, parameters, symptom change and quality of life, I will not dwell on that too much, but the problem there is that the subjectivity forces us to create tools again to turn the subjectivity into objectively measurable parameters. Therefore, we have the quality of life scores. Therefore, we have all those tools that you are likely frequently involved with that are trying to anticipate and trying to measure something that may be a perception and turn that into a reality that can be measured with scientific rigor. Still, the rigor of this is less, and symptoms or parameters like this are currently not accepted by the regulators for ultimate approval of drugs. Now, there are other measures where we're trying to measure drug activity, serum marker changes, tumor tissue, target inhibition measurements. You can compare tumor tissue to normal tissue as far as the target inhibition is concerned. Changes in PET scanning. There are many, 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 many. And unfortunately, none of them have, up until now, been able to be validated as a truly alternative endpoint. So they're not accepted by the regulators as a point of proof or evidence that something really works. So we are really still in need of other parameters that we can measure on a more easy way, and a less subjective way, to convince the regulator that something really does work. So I stated here in oncology, obtaining, oh, now I'm losing my whole, sorry, my PowerPoint goes out. That's something I had not anticipated on. Need to open it again. I apologize for this.
No worries. It's always technical issues are quite normal.
Yeah.
I think everybody has been used to that already during this last year.
Yeah. Now let's see, reasons. Takes a while. Doesn't happen too often. This way I run out of my time, that's a problem.
No worries.
Okay, let's go fast to where I was. So these alternative endpoints are not yet accepted by the regulators. And getting objective evidence is still a scientific art, is really art. If you go to the regulator, this is a chart that they will use on what they would accept as levels of evidence. There are different levels of evidence. One of the nice examples here is that a French doctor was believing that hydroxychloroquine was good against COVID, but that was a belief, and people are now using that drug. When they did a randomized controlled trial, actually there was no evidence of any effect, and if there was any evidence, it was against the drug. So currently the regulators are insisting on this level of evidence, so the randomized controlled trial. Now that randomized controlled trial is large and therewith expensive, involves a lot of patients, and always compares to the best therapy. So it's a comparator trial, always at least two arms. One, our patients get the standard of care. In the other arm, they get the new drug that may be added to the standard of care, or maybe a completely new treatment. This is a concept that people believe that was invented in the 1940s by Bradford Hill when he designed the very first randomized clinical trial in an antibiotic treatment of tuberculosis. But if you go back in history, the randomized trial was invented by a doctor on sea in 1474 when Dr. James Lind was faced with scurvy, which was a deadly disease among sailors, and decided to take 6 pairs of 2 sailors each with scurvy, and he treated them with, well, you can see it here, vitriolic elixir, vinegar, seawater, 2 oranges and a lemon, purgative mixture, and then was assessing how those people went. He was lucky or not lucky. Of his 6 pairs, 5 pairs died, and only 2 survived. And these were the 2 sailors that got oranges and a lemon. And we didn't know that vitamin C was the origin, vitamin C deficiency was the origin of scurvy in those days. Now we do know. But basically, this was already the first randomized trial that proved that something worked. Sometimes it's difficult to get evidence. There are some things that you cannot test. That's never been proven that the parachute works. There is no randomized trial of a parachute effect. And I don't think there will ever be a randomized trial on the effect of a parachute. So some things we will never be able to prove. Moving to the end of the story, rare diseases, we are dealing with sarcomas in this community here, the object or the subject of sarcomas. Rare disease, you all know that 1% of all malignancies are sarcomas, and don't even try to read those columns on the left side. That's the classification of sarcomas, and there are over 100 different sarcomas. So if we talk about soft tissue sarcomas, we basically talk about more than 100 different diseases. And how difficult that makes life is shown here. This is a study that was accepted for approval of the drug called olaratumab, a phase 2 study, not a statistically powered study. And you can see there's an impressive difference in the two arms. The yellow arm is the experimental arm. The problem was that the blue arm did worse than we had ever seen, and the yellow arm did better than we had ever seen. And then you start to disbelieve the effect. And if you look at the reality there, I mean, this was published, so people found this was very rigorous. I started to doubt it. And there are over 26 different tumor types per arm in each of the studies. So it was not a surprise to me, unfortunately, that the randomized phase 3 study that was used to prove the ultimate effect did not give any evidence of benefit of this drug called olaratumab. So basically it doesn't work. The other interesting observation was that in the phase 2 study, the drug was active in patients that not have had expression of the target of the drug. So a targeted agent that works in absence of its target is something you should really be critical upon. So we need that randomized controlled study, and it should be a phase 3 study of adequate size, otherwise the regulators will not take it. They will accept any evidence that comes from a phase 3 study. They will give conditional approval sometimes if they think there is a high unmet medical need, and if the benefit between risk and effect is positive. But that's usually a conditional and temporary approval and needs to be confirmed in a randomized controlled phase 3 trial. With diseases such as sarcomas, we are now also into a field which is making it even more complex. You will hear about basket trials or umbrella trials, which are basically trials that are focusing on genetic aberrations. An umbrella trial is a trial that uses tumors of the same type, and they do no longer use sarcomas as one tumor, but they will differentiate between the various types of sarcomas. And the basket trial is basing itself on a genetic change, because some genetic changes could occur in GIST, but just as well in gastric cancer or melanoma. These type of trials give us smaller data sets and are now being used by the regulators to temporarily give conditional approval of drugs for specific uses. And here's one example. Pembrolizumab, for instance, is used for patients that have microsatellite instability, high evidences in their tumor. That's a range of tumors. In colorectal cancer, this is very common. In those 149 patients, there were 2 patients with a sarcoma. Both of them did not have any response to the treatment. And yet, because the registration is based upon the total set, this drug is acceptable and approved for patients with sarcoma with a high macrosatellite instability. So this is basically muddy waters where we're still in, and not yet real solid evidence. So in summary, objective approval is difficult to get, but you can get it. The increasing knowledge that we have on diseases leads to further fragmentation of diseases, and everything is now turned into a rare disease, not only sarcomas, but even breast cancer. All the subtypes are becoming rare diseases, and rare diseases create another challenge for large trials. And that may be opposed, the scientific river may be opposed to the needs of the patients. And that's something we have to solve in a dialogue. And basically what I would like to end with is that we need a continuous discussion among all stakeholders, the regulators, the doctors that are treating the patients, the pharmaceutical industry that's developing the new drugs, and of course the patients themselves, the patient advocates, to find a common language and agree on what we accept as rigorous objective evidence. And that's where CDDF stands for. We are trying to get together the stakeholders that are in this debate, involved in this debate, and try to, within the rules that are there, try to navigate ourselves to the quickest possible availability of new drugs to patients all around the world. Sorry for running over time a little bit, but I'd like to thank you for your attention for this time.
No worries.